I'll start by introducing our first speaker today. So our first speaker is Dr. Zlata Gvozdenov. Zlata performed her PhD research work at the University of Illinois at Urbana-Champaign in the lab of Brian Freeman. She then moved to Harvard Medical School for her postdoc in the lab of Kevin Struhl. She's received several awards, including an F32 fellowship, as well as a fellowship from the Merck Foundation. Today, she's going to share her recent work distinguishing biological function from biological noise. So please join me in giving a warm, albeit virtual, welcome to today's first speaker, Dr. Zlata Gvozdenov.
Martin, thank you so much. Ben Martin, thank you so much for this kind and generous introduction. And I'll go through the talk now. I'm happy to present this little unusual topic titled Distinguishing Biological Function from Biological Noise on the example of transcription and chromatin organization. Some years ago, my advisor, Kevin Struhl, calculated based on the ChIP-seq experiments that 90% of elongating RNA polymerase II molecules do not produce traditional messenger RNAs that are used for protein synthesis. So 90% is a large amount, only 10% are functional in this sense. So what are all these transcripts for? And hypothesis is that the transcription could be very erroneous process, and these transcripts don't have any function per se, but are just something like a noise. And while this was hypothesized, this was never measured. Another example I have here is that a single gene can have a multitude of different transcript isoforms, and only for a small number of isoforms we know why they occur. For most of them, we are not sure why they're happening. And so again, hypothesis could be that this is due to imperfect transcription initiation or cleavage and polyadenylation process. And it could be, but we don't know, it's hypothesized. And now we have, I have a final example that disrupted or open chromatin organization can result in cryptic transcription initiation. And again, the question is why these cryptic transcripts occur. Is there any downstream role, or is just because there is a chromatin anomaly and transcriptional machinery could access? Hypothesis of this project is that proportion of the observed events can be classified into ones that do not have biological function and are biological noise. Sorry, I'm not sure whether you can see the title. It doesn't show to me. So biological noise, yes. Here, yes, oh, thank you. The assumption of the project is that biological noise can be measured with traditional assays by using functionally irrelevant DNA. What that means pragmatically is that how I did this pragmatically, introduced in the cells random DNA sequence artificial chromosomes and one chromosome, sorry, per cell, and then I measured molecular events such as transcription, transcript isoforms, chromatin organization, and I compared these events between random sequence non-functional chromosome and functional endogenous chromosomes, to see the capacity of the cell to generate noise and to estimate how much noise there is. Per definition, everything that happens at random non-functional chromosome can be considered as a noise because this chromosome is not needed. And just to make sure that we are on the same page in terms of the definition of biological noise or what is meant in this project, this is an antonym to biological function characterized by reproducible errors in the system which are not meaningful, and it is different from the stochastic variations within given cells. I'll run briefly through the strain construction. Overlapping DNA fragments, overlapping random sequence DNA fragments that were predefined computationally were synthesized, and then these fragments were co-transformed with so-called TAR vector that has yeast artificial chromosome elements. This was co-transformed into yeast, and relying on very powerful yeast homologous recombination, and the whole chromosome was assembled. Before, like, the main goal of this project is to measure transcription, but before I get into that, we wanted to make sure that chromatin is doing okay, because if there's too much chromatin exposure or even chromatin breakages, we wouldn't be able to have a fair comparison between genomic and random DNA. To look at the chromatin, I used RNAseq. RNase enzyme cleaves chromatin in such a way to release mononucleosomes, and there I wash away proteins, that is octamers or histones, and extract only mononucleosomal DNA for library construction and sequencing. Here I have a typical snapshot of the genome browser for a sample location where chromatin shows, or signal for the chromatin shows nice peaks that stand for nucleosomal positioning, especially at the beginning here of the gene. Unlike naked DNA control, that is RNase cleavage of the naked DNA, where no such profile is present. And then as a control, mitochondrial DNA chromatin under quotation mark and naked DNA look comparable because mitochondrial DNA doesn't associate with nucleosomes. And now the good news is that random DNA chromosome is readily associated with octamers to form nucleosomes because chromatin and naked DNA counterpart don't look the same. And the conclusion here is that random DNA is similar to genomic DNA in terms of the chromatin forming, and it is dissimilar to naked DNA. The positioning is different, and I looked at in the following experiment. I measured midpoint frequency. Midpoint here is a middle position of a DNA fragment generated by RNase cleavage. And so the more midpoints we have, the better positioning is expected over here. And then if we have less midpoints per nucleotide position, we're coming to randomly positioned nucleosomes, and very low midpoint frequency goes to nucleosome depleted regions over here. Chromosome 17 is actually our random DNA chromosome. It was dubbed chromosome 17 because there are 16 chromosomes in yeast, and simulated data here in gray is computationally generated positioning that happens purely by chance. And so random DNA does have some well-positioned nucleosomes, but little less than genomic DNA. And then random DNA has more randomly positioned nucleosomes than genomic DNA, about 50% less. And compared to genomic DNA, random DNA lacks nucleosome depleted regions. So as a conclusion, random DNA does have some well-positioned nucleosome, but random DNA lacks nucleosome depleted regions. I talk now about nucleosomal, one single nucleosome in their positioning, but what about nucleosomal arrays? We know that in the yeast genome, one or other genomes as well, one well-positioned nucleosome is followed by another well-positioned nucleosome and so on. This positioning slowly declines. The same is observed for the nucleosome depleted regions. For random DNA, this is not the case. So random DNA fails to form phase nucleosomal arrays. Now I'm coming finally to transcription at random in genomic DNA. For genomic DNA, we know, this is again snapshot from the sample region, we know that the transcription is organized. We have here plus strand transcription and minus strand transcription for one or the other gene. And in between, we have intergenic locations where transcription is lacking, how it should. This is a simple RNAseq or steady state experiment. And then for random DNA, surprisingly, transcription occurs throughout entire stretch of random DNA in both directions. And we lack these locations from which transcription is absent. Then I quantified transcription per 1 kb regions, both for genomic and random DNA. For genomic, I was really agnostic to regulatory locations, and I could see that the amount of transcription at random DNA is comparable to the average genomic transcription. I talked about the steady state transcription, but what about active transcription? To look at that, I used 4TU-seq, namely 4TU 4-thiouracil analog was spiked into the logarithmically grown cells from which I then isolated messenger RNA. And with specialized biotin, I purified away 4-thio-U containing mRNA. That is, I removed the rest of non-4-thio-U containing mRNAs. And that way, only quickly synthesized mRNAs that managed to incorporate 4-TU mRNA are proceeded with sequencing and analysis. I just mentioned that for the steady state transcription, the amount of random DNA transcription is comparable to genomic transcription. For active transcription, this is actually not the case. We have more random DNA transcription than genomic transcription. Now, comparing this to, because steady state levels are lower of random DNA transcription, we assume that degradation is higher, and this was checked in the following this equation that states production for TU-seq is proportional to the decay rate times the number of molecules, which is RNA-seq in our case. Here I derived half-life, unitless half-life estimator values, which showed us clearly that random DNA is enriched for shorter-lived RNA species, whereas genomic DNA is enriched for longer-lived RNA species. What about 3' isoforms at genomic and random DNA? I mentioned that a single gene can have a multitude of distinct transcript isoforms. These are 3' isoforms shown. There are different locations for the same gene where transcription terminates and then transcript is polyadenylated. We use a very modified RNA-seq that allows for identifying these features to identify single isoforms in a method called 3' reads. And so I use this to ask whether random DNA can be polyadenylated, can distinct isoforms occur, and is polyadenylation profile similar to genomic DNA? The answer is yes to all three questions. Here again, a snapshot from genomic DNA polyadenylation signal and for random DNA polyadenylation signal. Polyadenylation for genomic DNA usually occurs with an intergenic that is 3' untranslated regions here and over here. Whereas for random DNA, it happens pretty much throughout the entire stretch, is more comparable to this 3' UTR than to open reading frame locations. Isoform intensities are comparable between random and genomic DNA. And then cluster formation, that is the distances between adjacent isoforms, seem to be very comparable between random and genomic DNA. These isoforms tend to occur in clusters, as you can see from the snapshot. Here again, I have computationally generated isoform locations, again, that happen purely by chance, and they are more spread out. This is not the case for random DNA. Apparently, it behaves more like genomic DNA that happens in isoforms occur in clusters. And now, while these features are extremely similar, they're not identical. You can see that genomic DNA does tend to have higher intensity isoforms, and then genomic DNA isoforms do tend to occur closer together than random DNA isoforms. But we have more specificity. I won't bother you with quantification now, but it's about 10% of more specificity. When I go back to my first slide and the question, is there function or noise? I just show you there is a lot of transcription, and there is a lot of these transcript isoforms that happen on random DNAs as well. It's very likely that these are noise, just to reflect back on the first slide. And I looked at the nucleotide frequencies of transcript isoforms for top 10 isoforms according to the isoform intensities for genomic and random DNA. And you can see the same pattern. There is a little bit of more noise over here, but random non-functional DNA does form isoforms to the same comparable extent to genomic DNA and apparently to the comparable nucleotide frequencies. Final section here that I'll discuss is 5' isoforms at genomic and random DNA. I talk about 3' isoforms, that is different locations where transcription can end, but we equally have different locations for the same gene where transcription starts within 5' untranslated region. And one feature of these, of the start of the mRNA molecule is so-called 5' cap, which eventually protects mRNA from degradation. But this feature is used as a trick to identify locations where transcription started. Now, I improved on the previous methods to generate a more efficient approach and a faster approach to look at these isoforms, and this is now available in preprint. The method is called 5'-seq. So I use this to ask, can random DNA be 5' capped? Can distinct 5' isoforms occur? Is 5' cap profile similar to genomic DNA? And again, me with my snapshots from the genome browser of a sample location for genomic region, you can see high spikes within intergenic locations, that is 5' UTR regions, where we have accumulation of transcription initiation, how it should be. Surprisingly, for random DNA, this is not the case. So 5' isoforms behave differently than 3' isoforms, which were very similar to genomic DNA. We don't have such accumulation of transcription initiation per single locations or per single region. When I zoom in this signal about 20 times, you can see that there is a lot of transcription initiation all over the place for both genomic and random DNA. And if I exclude these high intergenic spikes, that is 5' UTR spikes, you can see that genomic and random DNA are pretty comparable in terms of transcription initiation. And we have some suppression, more suppression within locations, as you can see here, antisense transcription over here. But in terms of transcription initiation, we see very comparable profile, reflecting back again on the first slides that ask, is this a function or noise? Most of this transcription that starts all over the place within genomic DNA is likely to be a noise because it happens within any non-functional DNA as well, in this case, random sequence DNA. And I'll come to the conclusion now. Random sequence chromatin organization is similar to genomic chromatin, but random DNA nucleosomes are less well positioned and lack NDRs. Transcription on random sequence artificial chromosome is comparable to the average genomic transcription. Random DNA transcripts are prone to higher active transcription and RNA degradation. 3' isoforms on random sequence chromosome are arranged roughly comparably to genomic 3' isoforms. Transcription initiation is comparable to the non-5' UTR transcription initiation. High similarities between random sequence and endogenous chromosomes indicate lack of function and high biological noise. With this, I would like to thank to my advisor, Kevin Struhl, for really amazing guidance. I would like to thank to Zeno for help and the teaching of the software development. I would like to thank to my lab mates for reinforcing always productive discussion. I would like to thank to Kevin's funding and to my NHF 32 and Merck Foundation Fellowship. I would like to thank you for your attention, and I'll be happy to take any questions. I'm sorry for confusion in the beginning. I couldn't see my titles of the slides.
Awesome. Thank you so much, Zlata. That was great. I'll remind people, you can either type your questions into the Q&A box or click the raise hand button, and I can then ask, or I'll unmute you so you can ask your question yourself. But maybe I'll get started. I've got a number of questions. That was very thought-provoking. I was wondering, for the random DNA chromosome where you don't see kind of NDRs or well-positioned arrays, do you think that this is because they're not forming in any of the cells, or is it that across the population of cells they're all forming in different places? So when you do the bulk experiment, it kind of averages into kind of a relatively...
Yes. So it's later one. The number of actually nucleosomes that were detected per RNAseq were actually higher for random DNA because it doesn't have 60% AT locations that are getting lost, and that was though normalized. So the amount of nucleosomes were comparable according to normalization. And the later one is the answer. This is average of different cellular populations where nucleosomal positionings are different in every cell.
Mm-hmm. And then I wonder if you could expect, like, if I was following correctly, it looked like where transcription started was kind of more random across, but where transcription ended, you actually had some kind of more like pile up of 3' end reads.
Exactly.
And so why do you think that what do you think that tells us about how like transcription, like promoters are encoded versus termination sites?
Yes, so it's very true. It's very good, and it was a very surprising observation. Indeed, the transcription starts all over the place, and now we have transcriptional activators that help us, and some other regulatory location that help us localize transcription to the same place. But it's not only that. We have well-positioned nucleosomes and the whole evolved hierarchy of events that are at 5' UTR. For 3' UTR, it seems that 3' UTR, now I might mention this from evolutionary perspective, they can happen anywhere, but in the cells where the sequence is present, but in the cells that somehow evolved to be pushed towards the end of the gene. If that makes sense. Now, there could be some elements in the middle of the open reading frame that will be susceptible to polyadenylation, and it could be that was hypothesized in the paper, that is connected in a way to protein translation. Why doesn't it happen?
Thank you. Okay, so then we've got some questions from Felix Jonas, who says, very nice talk. Do you know something about the length of mRNA from the random DNA? And also, does the random DNA include some known DNA motifs like Tata boxes?
The length is, I have never mentioned length directly, but indirectly, we have 1 out of 64 codons for start codon, and then 3 out of 64 to be stop. So they're expected to be very short, but to have long untranslated regions. I did look for Tata elements, but I haven't seen any significant correlation of, obviously transcription is constant all over the place. So without Tata or with Tata box, it was the same, if that answers the question.
Yeah. Okay, and then Joseph Nasser has his hand raised. I'm going to allow you to talk. Joseph, you should be able to unmute yourself and ask your question.
Yeah, hi, thanks for this great talk. My question is sort of conceptual and sort of how you're thinking about the results from the the artificial random chromosome and going back to the the sort of genome in general. I mean, you started the talk with saying something like 90% of polymerases were not making functional mRNAs. I'm not really familiar with that reference, but yeah, I guess it's just like thinking about what there's all this transcription happening on the random chromosome, right? And how are you thinking about like all that is presumably noise under the definition that you gave, right? And so like what do these experiments like help you learn about noise as you've defined it on the sort of normal chromosomes, if that question makes sense?
Yes, I'll go back to the 5' and transcription initiation. And that's so initial experiment, the calculation that was done by Kevin, my advisor, on the ChIP-seq experiment simply involves, I think it's a very, very good review or opinion piece that estimated, if you have, this is say a gene, that we're looking at this gene over here, and all this is transcription initiation. And so all these are eventually transcripts that are produced, but only the ones that start here are the full length ones. And you see already here that the amount is very high. And now when the genome is very long, but 5' UTRs happen only at some locations. And so only the ones that start in 5' UTR in this case are going to be functional, going to be used. The ones that start here in the middle, all over the place, are likely non-functional because this profile is very similar to what we have here. So based on these, we confirmed that this hypothesis that was initially posed by my advisor in 2007 in this opinion piece is very correct.
Yeah,
I hope that makes sense.
Yeah, that's interesting. Yeah, that helped. Yeah, I mean, I guess I would say that like, just because the mRNA is not sort of full length doesn't necessarily imply it's not functional. It might, might do something that we don't we don't know, right? Something like that. But yeah, this idea of the sort of distribution of start sites on the random chromosome is almost like the null or distribution, or like almost like the background, something like that. And thinking about that as the sort of the noise level is quite interesting. Yeah, thank you. That helped.
Yeah, we know function only for some of them, like really only a couple of non-coding RNAs that we know, but I'd say we can count them on the fingers of a single hand. What about all the others that we don't know? Have we not yet discovered, or are these actually it was to this part, I would just like to add that for all the processes that I have examined so far, I haven't presented everything for the time constraint reason, but it seems like there is just about 10% more specificity for genomic DNA. For all the processes, random DNA is utilized to the exactly same extent as genomic DNA, really in all processes that we looked at. Now, there wasn't more, I think I understand your concern, like a direct experiment that would tell you that this is actually a noise. If you could think of a way how to do that, I'll be happy to do it.
I don't know. Yeah, no, this is very interesting. I get it now. I get it. Yeah. Cool.
Thank you, though.
Awesome. Thank you very much, Zlata, and everyone for your questions. And yeah, so thank you again for your talk.